Oh my goodness. So Lord forgive a girl have hobbies, cravings, or literally anything because I was trying to go home because I had a work meeting, but then my meeting got canceled and I wasn't looking at my notifications. And so I got there and nobody was there. And then I found out the meeting was canceled. So I was already out in town and I was like, okay, I'm hungry. I am craving French toast. I ate French toast. I go back home and I'm already prepared for my genetic stream and whatnot. And tell me what, I've been on the toilet for the last 45 minutes. This is terrible. And now I'm about to start work. So I don't even have a lot of time to talk about this, but I am going to make a lecture stream, the very first one of the semester. Yay! We're about to have our exams in like a couple weeks actually. So I want to get the genetics stream out of the way so I can move on to nutrition, psych, and anatomy and physiology too, which is what I'm taking this semester. So without further ado, I will move on and I am going to talk about all of this right here. So today I'm going to be talking about genetics, the intro for that, the history, basic concepts, and DNA replication. So let's get into it. So I'm not going to even go into what the definition of genetics is because you should know that already. So let's just talk about first the theories of creation. So there are a couple of theories, think about four. Yeah, four. So the first one is spontaneous generation, which means like just out of nowhere, humans just arose and now we're here. So one of the examples for that would be religion. So you know how like Adam and Eve, that story, how like they just boom, they appeared on the planet. So nothing else happened before that. It's just spontaneous generation of humans. The second one is preformationism. So this is when miniature versions grow into humans. So homunculus. There's a there's this theory of a homunculus, which is basically like a sperm human. It's like it's like head of a human, but then like the tail of a sperm, and it's basically like a tiny human, right? Like tiny human, and then it developed and it became bigger. So like I guess like we all started out as like gnomes or like fairies, and now we're here. So that's that second theory. The third theory is pangenesis. This is gemmules from organs shed and collect in reproductive organs, meaning like throughout our lifetime, there's like small particles of our organs. So, for example, like a small piece of my eye, small piece of my nose, small piece of my hair, and it all collects into my reproductive organs. And then whenever we reproduce, that's why our offspring looks just like the parents because there's small gemmules, like the small pieces of the organs have shed, and now that they're going to be passed on to the offspring. This theory actually follows blended inheritance and acquired characteristics. So blended inheritance is just like that you inherit traits from both parents, so it's like blended. And then the acquired characteristics means like a lifetime trait passed on to the offspring. So I would say definitely like for an animal, if it develops a shorter leg during one lifetime, then when it has offspring, it'll pass on the shorter leg to the offspring. And that has been disproven, of course, because you know, like if I accidentally cut off my arm, my kid is not going to have a cut off arm necessarily. So the last theory I want to talk about is epigenesis. Epigenesis? I don't know. But this is the embryo being formed by information passed down, and environmental affecting factors can decide whether or not these traits are turned on and off. And this is the one that we follow today because this theory is based off of just information being passed down and nothing physical. Okay, well, let's move on to contributions. This is all the people who have contributed to the field of genetics and have helped us to know a lot about what we know today. So, Levine, he differentiated between DNA and RNA and discovered the three nucleo nucleotide components. So the three nucleotide components is the nitrogenous base, the sugar, and the phosphate. So the main difference is between the DNA and RNA is the sugar. I think it's, I don't know if it's just the sugar or I could search it up, but I think it is just the sugar that is in it. So DNA is deoxyribose and then RNA is just ribose. So yeah, that's what he figured out. And then Kossel, he isolated nucleic acid from nuclein. So he basically kind of like did like kind of like the first step of isolation of DNA. So Avery, MacLeod, and Cardi, I guess. Um, okay, no, never mind. So Griffith, he discovered the transformative principle. So there was two strains of this bacteria or virus. He discovered the rough strain had a protective coating on the outside, and that was not. It didn't cause like death. I forgot what the word for it was. Let me go search it up down here in my notes. Oh my gosh. Not me doing this right now. Okay. Oh, it's a virus. Okay, so it was avirulent. The rough strain had a polysaccharide coat and it was avirulent, meaning it did not cause death. But the smooth strain did not have that polysaccharide coat and it did cause death, so it was virulent. And what he found out is whenever he killed this smooth strain, it actually became avirulent. But if he killed like the smooth strain with the heat and he combined it with the rough strain, the person was or like the test subject would still die. So that's the transformative principle, meaning even if you kill off a virulent virus or whatever, if you combine it with just the rough strain, it can still like transform back into the harmful version. So yeah, so that was basically how he kind of like was going towards the goal of like learning that DNA is a genetic material. Avery, MacLeod, and Cardi also kind of built off of this experiment and learned about like they basically proposed that DNA is genetic material, but Hershey and Chase they confirmed that DNA is a genetic material by making an experiment about radioactive bacteriophages. So basically, like they injected like DNA and whatnot, and like versus RNA, and then they found out that it was DNA that decided the genetic material. So Chargaff, he has two rules about the mostly about the nucleotide bases. So the amount of guanine is the same as is it cytosine? I actually don't know. But it's just like G and C and then A and T. So adenine, thymine are the same amount, like percentages in each species. And the amount of bases vary by species. So for example, like, you know, me versus somebody else or like a human or an animal, you know, um, the different percentages will be different. However, the ratios, the base ratios of G and C and A and T are going to be pretty much similar. Not G and A, but G and C, and then A and T separately. So Franklin, Rosalind Franklin, she looked into wet and dry DNA structures, how they're different, especially with B51. I believe that is a dry DNA structure, and she basically kind of like found that image and kind of like got to see the structure of DNA. And so basically, Watson and Crick was able to look at this picture. And from Franklin's work, they proposed the DNA double helix structure. So that's a big discovery, finding out the DNA structure. Kornberg discovered DNA polymerase, and then there's a Mendelssohn-Stahl experiment that proved that DNA is semi-conservative, and we'll talk about that right now, actually. So we're about to talk about DNA replication, okay? And there's three replication models. The first model is the semi-conservative model, which says DNA contains one parental and one daughter strand. So that's what Watson and Crick basically said, and also was proved by Mendelssohn-Stahl experiment. And the conservative model is both parental strands stay together after replication, and the daughter strands contain all new nucleotides. So, like for example, like like there's nothing taken by the parents. It's just like a completely new individual. Nothing is retained from the parents. The dispersive model is the parental and daughter DNA are interspersed in both strands. So I guess like the daughter I am not sure. So yeah, that's the dispersal model. I think it's like the so you know how the conservative is like everybody is their like different like strands or whatever, like parents here, daughter here, no mixing. That's like one extreme, and I think the dispersive model is the other extreme, where like everybody is like mixed up. The semi-conservative is like you have some stuff from your parents, but some of the like the other half of your DNA strand is completely new and like individual to yourself. That's what I think it is. Anyways, those are the three replication models. We DNA follows the semi-conservative model. It's proven by Meselson, Stahl, and Watson and Crick also supported that. So let's talk first, before we talk about DNA replication, we're going to talk about DNA. So DNA composition, it is composed of a lot of nucleotides. And nucleotides are composed of three things. First, a nitrogenous base, a sugar, and a phosphate. So we're going to talk about DNA specifically. The sugar is going to be deoxyribose. That's the sugar. And then they're going to have a phosphate group, and then a nitrogenous base. There are purines. I think that's singular ring. Okay, hold up. I actually have to search this up. Yes, I think no. Purines have double rings. So purines have double rings, and these are going to be A and G. And then pyrimidines are only singular ringed, and that is C and T. Let me look at something super duper quick. Okay. Yes. Let me go back. Okay. So yeah, A and G are purines, which is 2 ring, and then pyrimidine is C and T, which is only 1 ring. Okay, so that's nucleotides. So this is one nucleotide. It's got 3 components to it, and DNA just has a whole bunch of nucleotides that are bonded together. Okay. We're going to talk about 3 types of bonds. The N-glyco bond bonds base to sugar. So that's what keeps the base, the nitrogenous base, to the sugar. The phosphodiester bond is basically the backbone of DNA, and it links the 3' carbon of one sugar molecule to the 5' carbon of another molecule. So that's like basically forms like that. The middle, I guess, the middle part? No, it's not the middle part. It's like this, I guess, the sides. I don't know. And then the hydrophobic interactions is not a bond, but it's an interaction. So this is the tendency of bases to hide on the inside and stack on each other, causing the double helix and twisting. So I'm going to see if I can figure this out. I'm going to draw a DNA structure. Okay. So the N-glyco bond links the base to the sugar. So I guess that's like on a like miniature scale. So if this is the base and this is the sugar, basically this is what the N-glyco bond is, like the line right here. And then the phosphodiester bond, I guess, is the thick like outside of the DNA. And then the hydrophobic interaction, so you know how the bases are on the inside? So like if you're looking at DNA and it's like going in like this, you know, these are the bases on the inside because they're hiding away from the water on the outside. So they're hiding on the inside and they're stacking on top of each other, right? And that causes the double helix to twist because the hydrophobic interactions, they're trying to like go away from each other, but also go away from water. So that's what causes the twisting and also the bases to be oriented towards each other. Okay, so there's 3 types of DNA. There's ABNA, there's BDNA, and then there's zDNA. I'm going to discuss the differences between these 3 really quick. So aDNA is dehydrated. There's no water. And actually, let me look really quick. Okay, and then the bases are tilted, and then the R, it's right-hand formation, and then the configuration of the base is anti, and it's wider and shorter than BDNA. BDNA has water, the bases are stacked, and it is a right hand formation, and it's an anti-configuration. This is the most common type of DNA. ZDNA has a left hand formation. The bases are zigzagged, and they have both syn and anti-configurations of bases. It is narrower and more compact, and there are repeating purine and pyrimidine sequences. So you'll see like GC, GCG, or ATATT. I'm going to look up the 3 types of DNA. I'm going to look at the shapes, and I shall show you. Oh, okay. So let me draw this out. So it's easier to see. So this is gonna be A DNA. It's definitely wider and shorter, so it'll be like, whoa. I don't know, that's ugly. And then B DNA is what you think when you think of DNA. It's like pretty uniform, normal looking DNA. And then Z DNA looks like it's been on drugs. So it's like this. So A DNA is kind of like fat, and it's kind of like giving like if you're curled your hair or like you had wavy hair. And then B DNA is a nice little like curl looking like one of George Washington's wigs. And then Z DNA is like your hair when you first woke up in the morning. Okay, so there's that. And then let me show you like a cross section. So ADNA is going to be very circular. It's looking like a whole like ringworm, right? It looks like that. BDNA looks like the sun. So it's like that, and then it got some. It looks kind of like a flower, BDNA. And then again, ZDNA is on drug. It looks like a flower, but it's like a crazy looking flower. So yeah, that's what it is. So A is a ringworm, B is kind of like a sunflower, and then Z is like a flower on drugs. So that's how you can remember that. The main differences between each DNA: doesn't have water, B does have water, and then A and B are both right hand formation and anti-configuration, while Z DNA is left hand formation and both configurations. And then the bases on all 3 are different. A has tilted bases, B has stacked bases, and Z has a zigzag basis, which contributes to the shape that I just showed you just earlier. And then again, A is wider and shorter, and then B is just the most common, like regular DNA, and then Z is narrow and more compact. So that is basically the structure of the 3 types of DNA. We are now going to move on to the last part of this lecture recording, and I am going to discuss DNA replication steps. There's actually 5, but the 4 that Professor S discussed, I'm just going to discuss those now. So there's 4. There's initiation, unwinding, priming, and elongation. Let me X this out. Okay, so initiation starts from a replicon, and what a replicon is it's just a segment of like DNA where you will start the replication. So the replicon has an origin of replication, and this is where the initiator proteins will bind to the origin of replication. So it'll be this replicon. It has a origin of replication. Initiator proteins will bind to this site and recruit other proteins for preparation of the replication. After that, we're going to do unwinding. There's 3 enzymes that are involved with unwinding. There is helicase. This breaks the H bond between the bases and basically causes the unwinding. The topoisomerase or gyrase reduces torsional strain for helix unwinding, so it's not gonna like it's like mediating the craziness that's going on. And then SSB or single strand binding proteins, this stabilizes the ssDNA. And basically what this is it's let me read you this again. So the gyrase or topoisomerase, this reduces the torsional strain. You know how like the bases have torsional strain, and that's what causes like the DNA twisting. So if it reduces the torsional strain, what's going to happen? The twisting will not be there anymore, so it's going to straighten out. Basically what the DNA does is straighten out. Helicase is responsible for breaking those bonds. Topoisomerase is responsible for like untwisting, and then the single strand binding proteins, this stabilizes the single strand DNA to prepare for like replication. So basically what this does, it breaks the bonds, it straightens it out, and this creates a replication bubble. So this is what a replication bubble is. You don't see the bubble, whatever. This more looks like a little zipper. So, but this is the bubble right here, where the bubble is basically where it's unwinded, right? And then there's a replication fork. This is the replication fork. The difference between where it is unwinded and where it's not unwinded, that's where the replication fork is. And yeah, so the leading strand will synthesize towards the replication fork, and the lagging strand will synthesize away from the replication fork. That's basically what you need to know about that. And also, the double strand DNA, when it gets unzipped, that becomes a single strand DNA. So it'll be dsDNA, unzipped becomes ssDNA. And it's important to know that replication only happens for ssDNA. There's no way of like replicating a dsDNA because it's not unwinded or anything. So it'll just be like that. That's the dsDNA, this is the ssDNA because it's only one strand. Okay, moving on to priming. Primase and also DNA polymerase 1 or A will make short like RNA primers, because like DNA polymerase, there's multiple types, but I'm saying like in general, DNA polymerase is like important for elongation and like replication. But DNA polymerase has a huge fault, and the fault is that it cannot synthesize new nucleotides without something like without a nucleic acid already being there. So like for example, it's like a professor in a school. A professor can't teach a class unless the classroom is already there and the students are already there. So the primase, I guess, would be like the recruitment team at the university, or like the schedulers, the recruiters, the president, everybody involved in setting up a class, setting up the buildings so the teachers can teach. So just think of primase being like the recruiters in establishing the building, establishing the class, and DNA polymerase being the teachers. So they're really good at teaching and expanding knowledge, but they cannot do that without a class or a strand already being there. So that's basically what a primer is. So primase, it'll make short strands, like short RNA primers, for DNA polymerases to add nucleotides. So it can only add to existing nucleic acids. So these small strands are going to be 10 to 12 bases long, and I have drew them here. So these blocks right here will be the primers, the RNA primers for the DNA polymerase. And these are just only temporary for just like templates. These are going to be templates for DNA replication. Okay, we're going to move on to elongation. We're going to talk about a couple of polymerases. So in general, DNA polymerase will add new nucleotides to the 3' end of the RNA primer. So it'll synthesize from 5' to 3'. And you're like, okay, wait, why 5 to 3 when you said it's only adding to 3? Well, it'll start at the 5' end of like the entire string, like the entire strand, but it moves to the 3', meaning it'll only add to the 3'. If it adds to the 5', it means this synthesis is going to go to the 5' direction, which is not, it doesn't do that. It can only move one way, which is to the 3'. So it'll add to the 3'. But in general, the synthesis is moving from 5' to 3'. Okay, there's two strands being synthesized. There's a leading and then a lagging strand. This is for the daughter strand, basically. So polymerase E, I think epsilon, I don't know how to read Greek letters, so I'm going to figure it out later, but Pol E, it'll synthesize the leading strand continuously from 5' to 3'. So that'll be the one up here. The primer is already over here, so it'll add going this way. So this entire strand, this is the 5' end, this is the 3' end. On the bottom, it's opposite 5', 3'. So that means if it's synthesizing on the bottom over here of the top, like unzipped strand, it's going to move from 5 to 3. So you see how 3 is like on the bottom right here? So it's going to replace this bottom one, basically, and move from 5 to 3 continuously. Polymerase can only move from 5 to 3, which is why it is already in a good position here to move from 5 to 3 continuously. However, for the lagging strand, it is not. It is basically it'll move this way, right? Because this is 3 prime. So the primers are going to be added like a little bit more, like just in short segments. And so polymerase B, I think beta, or it looks like a B, synthesizes the lagging strand discontinuously from 5 to 3. And this will create fragments, and these are called Okazaki fragments. So basically, since this is the 3 prime, this is the bottom, the synthesis is going to replace the top. So it's starting from 5, and it'll move to 3, and then it'll start another segment here and move to 3, and then it'll like start another segment and move to 3. Since this is already going this way towards the fork, it's very continuous, but this is moving away from the fork. So it's like, where do I stop? You know, because like if you move from the outside all the way to the inside, you will know where to stop once the fork ends. But since this is moving away, it's kind of like hesitant. So that's why it's only moving in small fractions, right? So that's the lagging. That's why it's called the lagging strand. So Paul B will synthesize that in little Okazaki fragments until it's done. And then when it's done, all the primers are going to be removed by the polymerase, right? And then it'll be replaced by actual nucleotides. And then all of the Okazaki fragments are going to be sealed because there's gaps, there's gaps. These are going to be sealed by ligase. So that's the enzymes that will seal those. So I drew this. These are since the blue is the synthesizing, the black right here is the parent strand. So the daughter strands are going to look, it's a little bit confusing. Let me take you closer. Okay. So this is the parent. And so you see how this is the top of the parent that has unzipped. That means if this is 5, then this is 3. And if this is 3, like forget the bottom right here. If this is 3, that means this one has to be 5. So the synthesis is going to move from 5 to 3 until it reaches that replication fork. Okay? Now ignore the top one and look on the bottom. If this is 3, then this is 5, and the synthesis is going to replace that top strand right there. So it's going to be 5, 3. So it's going to start from here, move this way, from 5 to 3, from 5 to 3. So this is bidirectional, meaning it'll go this way. So this is going to move to the left, and this is going to move to the right. However, it's technically only moving in one direction. It's only moving from 5 to 3 in every situation. See, this is moving from 5 to 3, and then this is moving from 5 to 3. Technically bidirectional, but also technically only in one direction. Do you get it? So after all this is done, this is going to be the daughter strand right here. Or, this is going to be the daughter strand. 5 to 3, 5 to 3. This is going to be the bottom. This is going to be the bottom synthesized. And this one is going to be the top synthesized. 3, 3, 5, 5. Hopefully that is understandable. If not, I'm sorry, because I already understand, and it's not my job to make whoever's watching this understand. But I understand, so that makes sense. Okay, main topics to know. The enzymes. So initiator proteins, by the way, not all proteins are enzymes, so I'm just going to make sure you know every protein. Initiator proteins start the origin of replication. Helicase unzips topoisomerase, and gyrase reduces the torsional strain and basically unwinds the helix. The single strand bonding proteins will stabilize the sDNA. Primase makes the RNA primers. DNA polymerase, depending on the type, it basically adds new nucleotides to the primer. Polymerase E will synthesize the leading strand. Polymerase B will synthesize the lagging strand. And ligase seals the gaps for the Okazaki fragments. That's the enzymes. Replicon is basically this. It has this small part. It has a origin of replication, it is basically the origin of replication. So there's an origin of replication on the replicon. The replicon is a segment of the origin of replication, if that makes sense. And then replication happens on ssDNA only. Replication happens only from 5' to 3' end, even though it's bidirectional. And then replication is semi-conservative. Proof here: parent, daughter, parent's daughter. So the daughter strand will have the DNA of itself, like its own like unique mutations, errors, whatever. But it's also like taking all the stuff from the parent. Okay. And then bidirectional, we've already talked about that. Yeah, so perfectly 30 minutes. I'm glad I was able to get that done. I will go back on the toilet now and hopefully be able to make it to my shift okay. But yeah, thank you for joining me for my very first lecture stream. I'm glad I am able to get through all this, and I will start on the anatomy stream pretty soon. But yeah, thank you. Bye.